Welcome guys, and in this video I'll be giving you a brief lecture on the topic biomolecules from standard 11 biology. As we can understand, it is a very important chapter in biology and one of the most visible associations between organic chemistry and molecular biology. So without further ado, let's jump right into the video. Now, if we see the name itself and break it into two parts, biomolecules can be broken into two words, bio and molecule. Bio means life and molecule means molecules or chemicals. It means molecules or chemicals which are made within the life itself, which sustain life and which are responsible for giving life. These biomolecules are much required for the all-day metabolic activities we do in our day-to-day life. Now, before going further, I would like to introduce you to some terms which we may need throughout the biomolecules. First term is cellular pool. Cellular pool is the combination or collection of all the biomolecules and chemicals present within the cell. Now, within cellular pool, we can divide it into two parts according to its state of existence. One will be aqueous part and one will be non-aqueous. The aqueous part will remain suspended in the cell or dissolved, and the non-aqueous will remain deposited in the cell membrane, cell wall or chromatin. In biomolecules, we can also divide them according to their organic or inorganic nature into two parts. We can also divide biomolecules further on the basis of their complexity and size into two different branches. One will be simple or micromolecules and another will be complex or macromolecules. Now, simple molecules are the building blocks of macromolecules. Macromolecules are just the chained or joined structure of micromolecules. Now, let us introduce you to the term polymerization. Small units which can join to form big chains are known as monomers. When they join, they form a large chain. The chain is known as polymer. Now let us start the analysis of chemical composition of a particular substance. First we'll start off with organic matter, then we'll start off with inorganic matter. We take a piece of plant matter or flesh from animal matter, then we swiftly grind it with pestle and mortar in trichloroacetic acid. Then the so formed thick slurry is passed through a funnel covered with a cheesecloth. The acid soluble part filtrates out and is known as filtrate, and the left out part, which stays above the cheesecloth and is acid insoluble, is known as renterate. Part which stays above the cheesecloth is known as retentate. Now let us go to analysis of chemical composition of inorganic material. We take a small piece of plant matter or animal matter and dry it until all the water comes out. Then we burn it so that all the carbon there can be oxidized. Now all the carbon and hydrogen are oxidized to water and carbon dioxide respectively. What is left is the inorganic part, which remains suspended as ash. This ash, when subjected on different chemical tests, isolation techniques and extraction based processes, gives us the different chemical composition of different tissues, different parts of our body. We can also divide biomolecules on the basis of the importance into two parts, namely essential biomolecules and non-essential biomolecules. Non-essential biomolecules can be synthesized or acquired by our body or are required in minuscule amounts, but essential biomolecules are much needed biomolecules which cannot be synthesized in our body or are needed in a large number. We need to acquire them from outside the body. That's why they are called essential biomolecules. In terms of chemistry, we can differentiate compounds on the basis of the functional groups attached to them, like carboxylic acid, ketones, aldehydes, but on the basis of biology, we can differentiate them as different entities, like amino acids, nitrogenous bases. Let's start with amino acid. Amino acid is nothing but a substituted methane where one hydrogen remains, one hydrogen is replaced by carboxylic acid group, one hydrogen is replaced by amino group, and one hydrogen is replaced by some random R denoted group, which can be methyl, which can be acetyl methyl. Based on the R, we can define the physical, chemical, and structural changes of amino acids. And there can be up to 500 amino acids for the presence of this R group, but only 20 are applicable for the formation of proteins. They show differing structures in different pH because of the ionizable characteristics of their carboxylic group and their amino group. Now let us talk about lipids. Lipids can exist as fatty acids or glycerol or esterified as fatty acids and glycerols. Fats have a low melting point. Sometimes when a phosphorus group is attached with lipids, then they can be termed as phospholipids, the much required framework of a cell membrane. In some carbon compounds found in living beings, heterocyclic rings can be found. Nitrogenous bases are one of those. If found attached to a sugar, they are termed as nucleosides. If found attached to a phosphorus group also, they are termed as nucleotides. Major nucleic acids like DNA and RNA are made of nucleotides exclusively. Now let us study the structure of carbohydrates a bit briefly. Carbohydrates are the proximate principle of food, where the hydrogen is to carbon ratio is 2 is to 1. There are some exceptions of organic compounds which have hydrogen is to carbon ratio equal to 2 is to 1 but are not carbohydrates, and are carbohydrates but do not have hydrogen is to carbon ratio to is equals to 2 is to 1, like rhamnose. On the basis of the functional groups they contain, they can be divided into two parts. One is ketose and one is aldose. Aldoses have aldehyde group and ketoses have ketone group. On the basis of their structure, they can be divided into three parts. Monosaccharides, which do not give simpler sugars on hydrolysis. They are the simplest form of sugar and contain up to 3 to 5 carbon constituent chains. Another is oligosaccharides, which have from 10 up to 15 to 20 number of constituent carbon chains, and it can give simpler sugars on hydrolysis. Polysaccharides, the most complex form of sugar available in nature. It is not sweet and insoluble in water and gives large number of monosaccharides when it undergoes hydrolysis. So guys, that's all for today. This is the end of today's lecture. Do not subscribe my channel. Just give your feedback on how to improve the video quality. Bye.